DNA replication is the process where one double-stranded DNA is copied to form two identical double-stranded DNA. This process is an important step of the cell cycle. It is part of the S phase of interphase. Here is a closer look at how this DNA replication works. This is a double-stranded DNA. The top strand is 5' to 3', while the bottom strand, which is anti-parallel to the top strand, is 3' to 5'. The green enzyme here is topoisomerase.
Topoisomerase helps remove the stress from the torque of the helix shape and prevents the strands from snapping while the DNA is unzipped by the helicase. Helicase is made of 6 proteins and unwinds the DNA's double helix into 2 individual strands. Then, single-strand binding proteins, which are also known as SSBs, help keep the strands apart.
DNA polymerase cannot create new DNA. It can only extend the nucleic chain. Hence, in order to give the DNA polymerase a place to start, the primase catalyzes the synthesis of a short RNA segment called a primer, which is complementary to the DNA strand. Then, the DNA polymerase starts at the 3' end of the primer and synthesizes a new complementary strand. Then, the sliding clamp helps assist the DNA polymerase by sliding it down the DNA parent strand while it is synthesizing the new DNA. Because the DNA can only synthesize in the 5' to 3' direction, and the two strands are anti-parallel, the DNA polymerase synthesizes in different directions. Since it synthesizes in different directions, one strand is bleeding as it is continuously synthesizing down the DNA strand, while the other is discontinuously synthesizing and going back as new DNA is unraveled.
Then the RNase H comes in to remove the RNA primers and degrades them by hydrolyzing the phosphodiester bonds. The remaining gaps are filled in by DNA polymerase, and the DNA ligase connects the Okazaki fragments together.
DNA replication has thus been completed, as the original double strand of DNA gives way to two identical double strands of DNA.